Let's get started. Welcome to the inaugural research lecture in our research lecture series. A couple of housekeeping announcements. First, if you haven't signed in, please sign in. It helps to keep track of how many folks are here so we can advocate that this is important and should be continued. And second, put on your calendar if it's not already, the next research lecture, which is October 14th at noon. And that'll be Cecilia Rios Aguilar from UCLA, the school that need not be mentioned. So today our guest is Bradley Peterson. Dr. Bradley Peterson is the inaugural director of the Institute for the Developing Mind at CHLA and the University of Southern California. He's also director of Child and Adolescent Psychology at Keck. His research uses brain imaging technologies to understand the origins of neuropsychiatric disorders, mapping the constitutional and environmental influences that confer risk for illness or protect against it, trigger its onset or progression, compensate for its presence, or mediate effective treatments. Specific diseases that he studies include autism, depression, bipolar disorder, ADHD, Tourette syndrome, obsessive-compulsive disorders, schizophrenia, eating disorders, nonverbal learning disabilities, premature birth, and the effects of environmental toxins on brain development. Although publication rules differ between fields, nonetheless, his publication total of 280 peer-reviewed papers would seem to be large by the standards of any field. And he has mentored a dozen graduate students and medical students and 45 postdocs and junior research faculty members. He earned his medical degree from the University of Wisconsin in 1987, completed a residency at Mass General, an NIMH postdoctoral research fellowship at Yale, and he previously worked at both Yale and Columbia. So, very impressive resume. Thank you.
So thanks very much for the opportunity to be here. I hope I get a chance to meet a large number of you, hopefully all of you, and just talk about some common interests and learn from one another. This is a little bit of a complex title. It's Maturational Vulnerability, Neuroplasticity, and Intergenerational Transmission of Illness in the Developing Human Brain. Trying to think about, what are the connections that may be relevant to the School of Education. A lot of my work has focused on learning and memory processes, which are, of course, relevant to education. This second component, neuroplasticity, is really a learning and memory process. And so you can think of a lot of the things that we're going to be discussing, or I'll be discussing today, as looking at neuroplasticity, but through the lens of psychiatric illness and what we've learned about plastic processes in the brain. Learning and memory systems that seem to compensate for the presence of illness. And certainly these illnesses that we'll be talking about are very, very prevalent in the population. They're present in classrooms, and so teachers deal with this all of the time. In order to understand neuroplasticity and how we compensate for things, we have to understand what they're compensating for. And that's really maturational vulnerability, as well as intergenerational transmission of illness, because most of the illnesses that we study are transmitted across generations. And so we've learned a lot about that within our lab as well. Those are disclosures. I don't think I have any conflicts that would be relevant here. Neuroplasticity. So I just wanted to, probably all of you are familiar with this, but honestly I don't know your backgrounds, and so I thought it was important to talk about this at the very beginning. Plasticity is a structural reorganization, meaning brain tissue reorganizing itself to the extent that it's able to throughout development. Reorganizing the neural elements of the brain in response to developmental and experiential demands. So when we learn things, that's an environmental demand, it's an expectation, and through repeated exposures and learning, we actually do reorganize brain structure. It's a dynamic process that's superimposed on other major maturational changes during childhood, adolescence, and adulthood. And ultimately, it's mediated by the strengthening of existing synapses, the connections between neurons in the brain. And that strengthening of synapses changes the expression of genes. It literally turns on gene expression, and that expression creates new proteins or modifies the levels of existing proteins, and that thereby changes the synaptic structure, dendritic arbors, and literally the connections between neurons in the brain. And in fact, Eric Kandel at Columbia won the Nobel Prize for describing those processes, and it's been intensively studied in the last 30, 40 years. Neuroplasticity supports compensatory responses in the brain in children and adults with neuropsychiatric illnesses, and we're going to talk a lot about that in the coming hour. Our understanding of these compensatory processes also requires an understanding of what's being compensated for. You can't really understand compensation without understanding the underlying vulnerability. So if one part of the ship is tilting or listing, another part has to come in to right it, and you have to understand both of those in order to really understand the entire process. So here's an overall outline. We'll talk about maturational vulnerability and neuroplastic compensation in Tourette syndrome. It may sound far afield, but we've learned an enormous amount about vulnerability and compensation in this condition. It's also highly prevalent. I assure you, all of you have met and known people who have Tourette syndrome, even if you didn't know that they had it. We'll also talk about maturational vulnerability and compensation in ADHD youth, an even more prevalent condition. I assure you, someone in this room has ADHD or had it as a child. Your children, someone in this room will have children or nieces and nephews who have it, and it's certainly present in classrooms. And then finally, we'll talk about what we've learned of the intergenerational transmission of both vulnerability and resilience to illness in major depressive disorder, another highly, highly common illness in the population. So first, we'll start with Tourette syndrome. The clinical phenotype, the symptoms of this disorder involve rapid darting movements called tics. Some people call them habits. They're tics. They're rapid darting movements that involve primarily muscles of the face, neck, and shoulders, but can involve the torso, the diaphragm, and extremities in less commonly. Before these movements and sound, we've learned that there's a premonitory urge. There's a sort of discomfort in that body region that initially you're only dimly aware of it. That urge builds and builds and builds until you have to give into it and you move that body region. It brings an immediate sense of relief, but it's very brief, short-lived, and then that tension builds again. You can have a pretty good sense of what it's like to have tics, because these we refer to them as semi-involuntary movements, and we also have semi-involuntary movements like eye blinking. So if I asked you not to blink your eyes for the next minute, you would have a sense of that internal discomfort of what it's like to have to try to suppress these things. It requires a lot of attention, a lot of mental energy. Eventually, most of you would be able to suppress for a minute, but not all of you. And if I ask you to suppress indefinitely, you can't do it, right? Eventually you have to give in to that urge. There's a flurry of eye blinks. There's a sense of release, like, oh God, thank you. And then if you go back to try to suppress them, it builds and builds. So that's what it's like to have tics. Gives you a sense of that. So these are suppressible, just like your eye blinks are, but not indefinitely, semi-involuntary. There are comorbid, co-occurring conditions. OCD, obsessive compulsive disorder, occurs in 40 to 60% of kids in both clinical and epidemiological samples. This is thought to be a variant expression of the underlying vulnerability. So if you have the genes for these conditions, you can express them either as tics or as OCD or as both in combination. ADHD is also present in 50 to 60%, but this seems to be only true in clinical samples. It's not true in community samples. So that doesn't seem to be a variant expression. It's just that if you have ADHD, you're more likely to come to clinical attention and be diagnosed with Tourette syndrome. This is the natural history of tics and Tourette syndrome. The modal age of onset, the most common age is age 6. A lot of kids will have transient tics. My 2 oldest kids both had transient motor and vocal tics, and then they went away after a few months. If they persist, though, past a year, by definition they're called chronic. If you have both motor and vocal tics, by definition, it's called Tourette syndrome. If the history is the best predictor of the future, so the longer they've been present, the longer they're likely to stay. If they do stay past a year, they tend to increase in severity until about the age of 9, 10, 11, where they plateau, and then they gradually come down until about the age of 18. And after that, they're fairly stable. These are averages, however. There's a lot of spiking on an hour-to-hour, day-to-day, month-to-month basis around this overall rise, plateau, and decline. So it usually attenuates by adulthood. And one of the things we try to do is to understand what determines that severity in adulthood. Because if kids are getting better, really what we want to predict is who's going to be severe in adulthood so we can intervene and do things better. If they do develop OCD, it's not in the grade school years. It tends to be in adolescence. So the typical scenarios, tics begin in grade school, they increase, and then in adolescence, just as the tics start to come down, OCD blossoms. This is an overview of the maturational vulnerabilities and compensation we're going to be talking about. So this is the thumbnail sketch. I'm going to put a lot of meat on it in just a moment. But here we're viewing the brain from the side. Here's the forehead, back of the head, top of the head, of the left hemisphere. My laboratory, what you'll see is that the current belief is that there are massive circuits, parallel circuits that run from the cerebral cortex down to the basal ganglia here, these nuclei deep in the brain, and then back up to the cortex. These reverberating loops are very important in controlling thought, emotion, and behavior. The control of motor behavior typically comes from somatosensory cortex here and loops down the basal ganglia and back. Our data and other laboratory data suggests that these loops are hypoplastic. They don't grow sufficiently in kids who have Tourette syndrome. So they're reduced in volume, in anatomical images here in the sensory motor cortex, the basal ganglia, especially caudate, and other portions of these loops from cortex to subcortex. So that's the underlying vulnerability that these kids come into life with. It's a hypoplasia of these motor circuits. In a lot of kids, not all kids, but a lot of kids, they're able to kick in compensatory processes, not in the sensory motor loops, but in these loops that run from frontal cortex down to the basal ganglia and back again. So literally they are able to change brain structure in response to the presence of these unwanted behaviors, and especially by trying to control them, they increase the volume of the frontal lobe, and to the extent that they can do that, it attenuates their symptoms, not only of tics, but also OCD. There's another structure here in the temporal lobe called the hippocampus. So sitting on top of it, or on the front, is the amygdala. In blue here is the hippocampus. This is connected to the frontal lobe. The two work in concert. And this also seems to hypertrophy in kids who have Tourette syndrome. And it works in concert so that the degree to which it increases in size and volume, along with the frontal lobe, it also tends to reduce symptoms of tics, OCD, and also ADHD. So I'm going to disimpact this in the coming slides, but that's the overall picture and theory of pathogenesis and compensation. So hypoplasia, or sort of insufficient growth of these sensory motor circuits in Tourette's. So this comes from a large sample of kids and adults that we've studied. 155 Tourette's kids and adults, those age range. Normal controls 131 in the same age range. This data set represents imaging at only one point in time, so it's temporal cross-section. And I will say, just now, so it's a sort of disclaimer, if you eliminate anyone who has OCD or ADHD, the findings are exactly the same. If you remove anyone who ever has had medication, the findings are exactly the same. So the findings are not from those other illnesses or medication. So this is a paper from Nature Neuroscience a few years back. And let me just explain what you're looking at. So here's the left hemisphere, forehead, back of the head. Here's the right hemisphere, forehead, back of the head. At each point across the surface of the brain, literally millimeter by millimeter by millimeter, very, very fine-grained anatomical measures of the thickness of the cortical mantle. So the cortical mantle is about 6 millimeters thick on average. It contains most of the nerve cell bodies and the vast majority of the synapses, the connections between nerve cells. And so that's where all of the computational work of the brain occurs, is that in the cortical mantle, by and large, especially conscious computational work. And so what we're doing is comparing the Tourette syndrome people with the healthy control people at each point along the surface in terms of the thickness of that cortical mantle. If you see something in yellow or red, it means that it's significantly, very significantly reduced in thickness in the Tourette group. And so where you see the red and the reduced thickness is here in the Broca's region, which controls language and speech output. In the inferior portion of the motor strip here and the sensory strip here. And you see it especially prominently on the right side in Broca's, sensory strip, and motor strip. You see some other stuff back here that we're not going to focus on just right now. We can also, within the Tourette group, we can correlate that thickness with the severity of their tics at the time of scan. And you can see here in red that the thinner the regions are here, higher up in the brain, or dorsal, in the sensory and motor strip, the thinner that is, the more severe their tics are. So across the whole sample, we have reductions here in red, and then for those who are most severe, we have more involvement higher up in the brain. So why is that relevant? Well, it turns out that those regions of the brain control everything that produces tic symptoms. So this is called the human homunculus. So this could be the motor strip or sensory strip. These are the portions that control various bodily regions. So along the medial wall here is the foot, hip, trunk, and then hand and face have much larger spatial representation because of all the fine muscles, right? So you have very large representation of hand, but very large representation of face, tongue, and larynx. So all of this stuff on the lower portion of that homunculus, of that motor strip, controls the face and vocal apparatus, right? And so these portions here correspond to these portions here. So you have thinning of the cortices that produce these abnormal movements and sounds. Then, if you have additional thinning up here, you have more severe symptoms. And what that really means is that you have more involvement of other body regions other than face and vocal apparatus. So you end up with higher ratings of severity. So that's at the level of the cortex. We've studied this entire loop, but I'm only showing you two sort of representative regions. Right here, we're looking at the caudate, the basal ganglia, which for a long time we thought were involved in Tourette syndrome for lots of various reasons. These tend to be involved in movement disorders. And so here's the brain rotating. The caudate nucleus is in purple, the putamen is in green, and nestled inside is the globus pallidus. Those are the basal ganglia. So we studied each one of those regions in terms of their overall volume. And we found that, in general, it was the caudate nucleus that was reduced in size about 5% or so. The other two nuclei were okay. There weren't volume reductions. It was the caudate specifically. And then we can do the same thing on the caudate like we do on the surface of the brain, millimeter by millimeter, to compare healthy controls and Tourette's. And this 5% volume reduction is coming from the anterior portion, specifically of the caudate. And that's what connects the caudate to the frontal lobe in terms of, and essentially controls information flow through the caudate. So what that's saying is that, at least in these two nodes, and I can tell you in all the rest of the nodes along this circuit, you have reduced volumes. So it's hypoplastic throughout the entire cortical, subcortical circuit, especially in sensory motor regions and pathways. The caudate nucleus and basal ganglia at the time of scan didn't predict very much in terms of severity. It just seemed to be a reduced volume, and the severity was sort of independent of that. Turns out, though, that we followed up this sample. So the kids who had been imaged before 14 years of age, we followed up now when they were 18. And we asked how their clinical symptoms were. And it turns out that the caudate nucleus volume in childhood predicts the severity of tic symptoms in adulthood. So you remember the tic symptoms increase, they plateau, and then decline. And we have no clinical predictors of who's going to have severe symptoms and who doesn't. This is really the most robust predictor we have, which is volume of the caudate. And so here it is on the x-axis, and this is adjusted for overall brain size, and here's tic severity on the y-axis. And so the smaller the caudate is over here, the more severe the symptoms. The more normal or larger the caudate is, the fewer the symptoms. So the degree of hypoplasia, the degree of this maturational vulnerability, seems to influence where tic symptoms are in adulthood after the flux of development settles down. The really interesting thing, in my opinion, is that caudate volume is also correlated with OCD symptoms in adulthood. So in childhood, none of these folks had OCD. A lot developed OCD symptoms and disorder in adolescence, and it was exactly the same relationship here, so that the smaller the caudate, the more likely you were to develop OCD. So it's predicting the appearance of a disorder in adolescence that isn't even present yet at the time of scan. This is a little bit dense. I'm just going to fly through it a little bit, if that's all right. But it turns out that those hypoplastic motor circuits, cortical, subcortical circuits, that haven't grown properly in development are, for some reason, hyperexcitable. There's too much activity in them. We think we have ideas about why that is. The inhibitory neurons seem not to be present or as active as they need to be, so they're not inhibiting activity in that circuit. But here's some of the data for it. What we had these folks do, we had adults with Tourette syndrome come in, just lay in the scanner, and we imaged them with functional MRI. And we noted when their tics occurred throughout the scan. We had healthy controls come in, and they practiced mimicking a facial tic. So they became very good at it, and you could distinguish it from a real tic. So we're looking at the activity in the motor pathways, at each node along the motor pathway, in real tics in Tourette syndrome, and then mimic tics in healthy controls. And now we're going to see, the question is, do tics engage the same motor circuit as normal voluntary behaviors? Now that may seem like a silly question, but it was a hot one in the field. People thought that they weren't actually engaging motor circuits, that these were sort of escaping behaviors generated somewhere else. So it turns out that, in fact, all of the nodes do activate in real tics, as you see in mimic facial tics. All of these nodes activate, but they activate more strongly in the Tourette group. And this is just going to show you some of the data. So these are two-dimensional slices of the brain, parallel to the floor in a standing person, so they're like this, right? So this is low in the brain, higher, 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 near the top of the brain. And here's the forehead and back of the head. So this is one node, which is the caudate, which you've already seen in our anatomical studies. Here's the globus pallidus, putamen, motor cortex, sort of motor planning cortex. Here are mimic ticks in the healthy controls. You can see the activity in each of those nodes. Here's in spontaneous ticks, you have activity in each of those nodes. But it looks stronger here. You have more intense red and yellow, indicating stronger activity. And when we compare those two directly, we confirm that at each of these nodes, the activity is stronger in real ticks. So they seem to be hyperexcitable. There's more activity in them, generating the same behavior. Actually, if you don't mind, we're just, but remember, and we'll come back to it. In the Tourette group, it turns out that the more activity you have, so on the y-axis is activity, and so tic severity is on the x-axis. So the more activity you have in these motor circuits, the more severe the symptoms are. There's an exception in the caudate and anterior cingulate, so frontal cortex and caudate, and it goes the other way, so that the more activity, the fewer the symptoms you have, and we think these are control regions of the brain. So this is frontal cortex, anterior cingulate, and so that when control regions are more active, you have fewer symptoms. That's going to be a recurrent refrain throughout this. So here's a summary of the circuit-based vulnerabilities. You have hypoplastic, meaning they don't grow enough tissue in development in motor portions of these circuits. They're also hyperexcitable, and they produce more activity during spontaneous tics than in normal voluntary behaviors. And then you have reduced activity in the caudate and other control regions that produce more severe symptoms. So motor activity is disinhibited, it's poorly regulated, and it's in direct proportion to the severity of symptoms. So that's the maturational vulnerability in this disorder. And I assure you that the rest of ADHD and depression are going to be faster. But it's going to give you an overview of how we go about this. So what about compensation in Tourette syndrome? It turns out that if you look at the surface of the brain, here we're looking at local volumes, millimeter by millimeter across the surface of the brain. Before, we looked at the thickness of the cortical mantle. This is looking at overall volume at each point along the surface. And this is comparing the Tourette group versus the healthy controls. And in regions where you see this sea green, it means the two groups didn't differ in local volume there, in the amount of brain tissue, if you will. In regions in red, you see increases in size in the Tourette group. You see it in the frontal lobe and then parietal lobe. So here's a view directly from the front. You see the degree of enlargement bilaterally. And this is a view from the top. So here's the front and back, and you can see the enlargement. So that's interesting. That's an unusual finding, is that you have a larger portion of the brain in a disease group. Usually it's smaller across most studies. So the question is why? What is that larger volume? And it turns out that it seems to be helpful. So here's a map of a graph of tic severity on the x-axis. This is volume of the frontal lobe. And you have this inverse relationship, so that the larger the frontal lobe, that is, the more abnormal it is relative to healthy people, the fewer the tic symptoms you have. This is true in kids and adults, both. So that larger volume seems to be potentially a helpful thing. This is another somewhat complicated idea, but it's important, and so I decided to show it. It turns out this frontal lobe here, and, on the y-axis, in the Tourette group seems to decline with age in the Tourette group. In the healthy controls, there isn't much correlation with age. So remember, these images were only acquired at a single time point, so we actually didn't take the same people and image them from age 5 to 54. Right? So these are all different people. Each of these dots is a different person. In imaging studies in the past, if you see correlations like this that differ across the two groups, the tendency would be to say, well, you know what, Tourette's is probably a degenerative disorder. You have a decline with age in Tourette syndrome that you don't have in healthy controls. That doesn't seem to be the case. This is not a degenerative disorder. Cognitively, they don't get worse or anything else. what we suspect instead, and I'm boiling down a lot of our data and other lab data, is that the overall enlargement of the frontal lobe is coming from the kids of the sample. So the kids of the sample tend to have larger volumes than the healthy controls, and you can see that. But there's a lot of variation within the group. So in green are the Tourette's kids. You can see all the scatter, and underneath it in orange are the healthy controls. The adults out here are generally, who have Tourette's in green, have smaller volumes than the healthy controls. And we don't think that if you take all of these kids that they, in the end, follow this trajectory. We think that these adults with Tourette's started out down here in childhood as having smaller volumes. And in fact, these kids, when they're followed, if you have smaller volumes here, you'll tend to have more persistent illness in adulthood. And then you end up into our imaging study. The kids up here, if you were to follow them, they're lost to clinical follow-up because we think their tics get better. And so they don't enter a study like this because they disappear. They're better. So that's what we think is happening, is that the adults that we sampled and brought into the study, they were samples of convenience from our clinic. We think that they had smaller volumes, and that's why their tics were still present in adulthood. Because remember, it usually declines and gets substantially better by age 18. So why would that frontal lobe enlarge in Tourette syndrome? Why is it enlarged? So this is one of our first fMRI studies back in 1998. And actually one of the first in the field studying, it was the first fMRI study in Tourette syndrome. What we had people do was we brought them into the scanner. We were interested in understanding the regions of the brain that control tics. And so we had them allow their tics to happen just spontaneously, not force them, not inhibit them, just let them happen for about 20 seconds. And then we would ask them to stop and inhibit them, just like I would ask you to stop your eye blink. And they could all do that. We made sure they could. So they would go back and forth between letting them happen and inhibiting them. And so when you compare brain activity in those two conditions, you can tell what brain regions are turning on when you stop the behavior. And it turns out it's all of these massive frontal activation in red. So it's frontal, 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 frontal. It also has some anterior temporal and then parietal. Those were the regions that were increased in volume in the anatomical scans. It's right where these kids have to generate activity in order to inhibit these tics. So these kids, especially kids during school time, even if they're not consciously suppressing them, it's the social awareness, even unconscious by habit, they have to exert a lot of energy and attention in keeping these things under wraps and under control. Once they come home from school, the typical story is, teacher said they was fine, they didn't even notice tics, and then they come off the bus and they're going wild, and just like you would if you let your eye blinks happen after suppressing them. And so all of this activity, this turning on of activity in the frontal lobes and parietal, we think that is what produces this hypertrophy of the frontal and parietal lobes. It's just like a muscle. The more you use it, the more you can increase the structure that supports that activity. There's this other structure, the hippocampus, that I said is directly connected to the frontal lobe. Now this is a structure should be near and dear to your hearts. It's involved in learning and memory in a very key way. And it supports a lot of the activity of the frontal lobe and processes of the temporal lobe. And it turns out that in Tourette's, it too is larger, just like the frontal lobe. So it's again a very unusual finding. It's about 5% larger in the Tourette group overall. And if you do this millimeter by millimeter comparison across the two groups for the hippocampus, just like we did on the surface of the brain, we can compare both. If it's green, it means they're very similar in volumes. If you see red, it means it's increased in size. So this 5% increase isn't coming from the whole structure. It's coming from this portion, the head of the caudate and medial portion of the tail. You can see it here on the undersurface. That portion of the did I say caudate? I meant hippocampus. So this portion of the hippocampus, we know from lots of animal studies and other human studies, this is the part of the hippocampus that undergoes plastic change in response to learning. So we think that this enlargement is very much like the frontal lobe, increasing in size in response to need. Over here you see maps of, not comparing the two groups, but within the Tourette group, we can correlate local volume at each millimeter of the surface with the severity of symptoms. Purple means they go in, it's an inverse relationship. So the bigger the volume, the fewer the symptoms. And so you can see for tic severity, it's in the region of enlargement here and here. And you can see on the undersurface, the regions that are enlarged relate to severity. So that the larger the region, the fewer the symptoms. The really amazing thing is it's not just tics, but it's also OCD severity and ADHD severity too. It seems to be a very broad modulator of control and self-control, so that when you can increase this in size, it helps to control all the symptoms associated with Tourette syndrome. So here's an overall model of pathogenesis for Tourette's. We think that kids and adults need to suppress these tics. When you activate the frontal cortex or parietal, you can generate a plastic response in both frontal cortex and hippocampus, right? That's neuroplasticity. If you're able to do that, then you end up with larger volumes. And we see that especially in our sample of kids. You end up with fewer symptoms, and I haven't shown you, but you end up with normal activation of the brain. For some reason, some people seem not to be able to either activate frontal cortex or generate this plastic response. We don't know why not. They just seem not to be able to do it. They end up with smaller volumes that we see especially in the adults. They end up with more severe and more persistent symptoms throughout life. And they have to increase brain activity much more in order to maintain normal performance on tasks. Okay, so that was a fairly detailed presentation of Tourette's, and we're going to march much more quickly through ADHD and depression. This is a paper of ours from Lancet in 2003, where we looked at cortical volumes in ADHD compared to healthy controls. And regions here in red and yellow were reduced in size in the ADHD group. So here in the right hemisphere, you see an anterior temporal, inferior frontal. And it's pretty symmetrical. You see it in the left hemisphere as well. The regions in blue don't differ across the two groups. So these regions we think are involved in attention and impulse control and control of hyperactivity. And so it was the first report of that at the surface of the brain in ADHD. That's why it got into Lancet. That was a cross-sectional study. And a group at NIMH undertook a longitudinal study. This is a considerably larger one. There were 223 kids with ADHD, 223 healthy controls. And let me just walk you through what these images are showing. It's really fascinating. So this is a movie that's going to loop. And what they are plotting here is at the surface of the brain is, as you see this kind of intermediate gray, when they feel that that cortex is mature, they color it in that intermediate gray. If it's not mature, it has this sort of white or light gray appearance. And then the very dark gray here were regions that they couldn't say definitively when that cortex was mature. So it's sort of, you have to mask that out, those very dark regions. So what you're seeing here, when it loops around, is that the typically developing kids mature much faster. So you'll see the frontal cortex and parietal and anterior temporal mature much faster, and sort of in a back to front pattern. So you see it maturing in the back and progressing to the front. And then ADHD is delayed by about 5 or 6 years on average compared to the healthy controls in terms of the maturation of these frontal and temporal cortices. So we, in our paper in Lancet, we thought that that was probably a static deficit, that they came in the world by age 6 with a certain abnormality and that it was the same by age 18. This says it isn't. This says that it's a maturational delay in the ADHD kids compared to the healthy controls. Let me just explain how that works. So it turns out that the cortex thins with age, and that's the healthy control here. So here's volume or thickness of the cortex, and here's age on the x-axis. Healthy controls are delayed by about 5 years. ADHD kids are delayed by about 5 years relative to healthy controls. At each point, at each age, say age 6 or so, the difference is a certain amount here on the vertical distance between these two parallel lines. That vertical distance is about the same at age 18. And so if you're imaging these kids only once, you'll see a constant difference across age. It appears to be static, when in fact it's a maturational delay. So that in our data set, when they covary for age, this is what it looks like. It looks like it's a constant difference across age. So the lesson here is that in order to really understand a developmental process, you have to do longitudinal studies. You can't infer safely longitudinal trajectories from cross-sectional data. So you remember in Tourette syndrome that the hippocampus seemed to hypertrophy, to increase in size as a neuroplastic response to the presence of symptoms. And that seems to be true in ADHD as well. It's 7% larger overall in the ADHD kids. And it's not everywhere, as you can see. The enlargement is in red here. So again, kind of the head of the hippocampus, here's the head and the tail. Very, very similar regions as in the Tourette group. And it turns out that if we correlate in the ADHD group the severity of symptoms with these volumes, you see these purple regions indicating the larger the region, the fewer the symptoms. That's exactly what we saw in Tourette syndrome. And we're seeing exactly the same process in ADHD. So the hippocampus seems to be crucially important in this neuroplastic response in controlling and modulating symptoms for kids who are struggling, attending to class, and staying seated and not blurting out and doing other unwanted things. This is a crucial structure. In the interest of time, I'm not going to show this, but that is showing us what the stimulant medications seem to do in terms of brain structure. They have a powerful effect that I can review later if you wish. Lastly, and very quickly, what about intergenerational transmission? So Tourette's is a highly familial condition, strongly genetic. ADHD ditto, very, very strongly familial, a lot of genetic loading. And major depression is as well, it turns out. So I've been very fortunate in collaborating with people at Columbia, Myrna Weisman in particular. She has a 3-generational, now 4-generation cohort that she's followed for more than 30 years now. I'm going to describe the first 3 generations and just need your attention for 30 seconds so that you understand the design and how we identify what's being transmitted across generations in the brains of people that predispose to becoming sick. So at least this is what's being transmitted in depression. It's not necessarily the same in Tourette's or ADHD. So in the first generation, in and around New Haven, she found a group of very severe and very chronically ill, depressed adults. She put together a cohort of never depressed community controls. So she interviewed parents and spouses, and apparently these folks had never been depressed by history. They had children, that she followed in childhood and adulthood in generation 2. Those people in turn had children. A third generation that she followed from birth into adulthood. And we imaged the entire sample, but I'm only going to show you results for generations 2 and 3, because we're interested in what's being transmitted, not in what is already present when things are, when people are already ill. So we're looking at biological offspring of generation 1 depressed and generation 1 never depressed. These biological offspring of the depressed people are at high familial risk for developing depression, and those offspring of the never depressed are at low risk. It doesn't mean zero risk, it means low risk. Some do develop depression. What we do then is we compare images in the high-risk offspring, generations 2 and 3, with images of the low-risk offspring. And what we're trying to understand is what's being transmitted in this group that's different from this group. I should point out that this same study actually did show, it was one of the first to show that depression is familial, so you get much higher rates of depression here. And it also was the first to really identify the natural history. So this group tends to become sick in adolescence and young adulthood, and it's much more severe and chronic than if you're depressed without a family history. So pictures are worth 1,000 words. Here's essentially what we found. So these are measures of cortical thickness. We already talked about that in Tourette's, right? The cortical mantle, where all the synapses are. And this is comparing, again, millimeter by millimeter, the high-risk offspring with the low-risk offspring. If you see this thing in this color in sea green, it means they didn't differ in thickness. If you see purple, it means it's dramatically thinner in the high-risk offspring. And so here's the right hemisphere. You see massive thinning from the back of the brain to the front and the top to the bottom. It's essentially the whole lateral aspect of the brain is thin in the high-risk group. You don't see it on the lateral surface of the left hemisphere. Here's the forehead, here's the back of the head. It's pretty clean on that hemisphere. So this is highly asymmetric that's being transmitted across generations. If you take the brain and split it right down the middle, here's the right hemisphere, here's the forehead and back of the head, here's the left hemisphere, forehead, back of the head. You see, so this is the opposite hemisphere. So this was right. You see thinning along the mesial wall from the back to the front in the left hemisphere. In the right hemisphere, you don't see thinning, you actually see some thickening in important limbic or emotion regions here and in the back here. This is one of the largest biological effects reported in an imaging study, and it involves about a 30% reduction on average in thickness in that right hemisphere. That's a massive effect at each point of the brain, and then it's massive in its spatial extent. So you may say, yeah, but we know that some of those offspring, those biological offspring, had depression. So they were followed for 30 years. We know exactly those who did have depression or not. And you're right. So that could have been driven by, that finding could have been driven by those who had depression. So we removed everyone from the sample, from both samples, high and low risk, whoever had depression in their lifetime. And this is what we found. It's exactly the same. It's the same finding. So these are people who had never been depressed in their lifetime, and they still are carrying this risk marker. It turns out that, so there's a lot more I can elaborate about this, but this is somewhat relevant to teachers, I think. So even if they've never been sick with depression, if they have this marker, to the extent that they have it, they have some cognitive problems. They have trouble with attention, sort of ADHD-like attention, but more importantly, they have trouble with learning and memory. But it's very specific kinds of learning and memory. It's learning and memory for social stimuli. So they have trouble remembering faces, social situations, social stories and narrative. And so we think that this creates a cognitive problem that sets people up for depression because they're sort of cutting off from others socially. This finding, by the way, was present in the youngest of kids. You can see it in individual scans, even by the age of 6. So it seems to be, the brain architecture is established very, very early, and this marker is easily identified in the youngest of kids. I'm not going to spend much time on this, but we also looked at brain function and in the same sample. And one thing I just want to point out was we were able to disentangle risk effects like I just showed you. So what makes you vulnerable? These are risk effects. These are sort of the scars or residua of having been depressed in the past. More importantly, though, we were able to identify resilience effects. And so there are portions of the frontal lobe, anterior cingulate, and other portions of frontal lobe, and here the prefrontal cortex. The more active those are in the functional scans, the more it seemed to protect the high-risk group from becoming sick. So it's very analogous to what we were seeing with ADHD and the importance of frontal lobe and vulnerability, but also the plastic effects of frontal cortex, anterior cingulate, in compensatory responses in Tourette's, and here it seems to be, in terms of functional activity, it protects you against becoming ill. So it's a resilience effect. So here are the overall conclusions. Again, looking at learning and memory and neuroplasticity through the lens of psychopathology. In Tourette's, we have a maturational vulnerability that's hypoplasia and hyperexcitable sensory and motor portions of the cortical subcortical loops. Neuroplastic compensation involves hypertrophy of frontal and parietal cortices and the hippocampus. ADHD, the maturational vulnerability is different. It's delayed maturation of frontal and temporal cortices. So it's a unique vulnerability. It's not the same one. That's why you have ADHD rather than Tourette's. But neuroplastic compensation does have some overlap. It involves hypertrophy of the hippocampus and the portion that I skipped over. In depression, the intergenerational transmission of vulnerability involves massive thinning of the cortical mantle in the right hemisphere. And then developmental resilience and plasticity seems to involve frontal cortex, both the anterior cingulate and dorsal prefrontal cortex. So again, overlapping systems for compensation and neuroplasticity. The final consideration is that identifying these neuroplastic circuits that support compensation and resilience provides targets for novel intervention and prevention. So we know now what we're learning what is really important in helping these children. And it provides a target. So are there ways that we can go in and modulate activity, support plasticity and growth, and reduce these symptoms and their overall functional capacity in classrooms, for example? This is just one cross-sectional look of the lab. If you multiply that by 25 years, you can get a sense of the people who contributed to this work. So thank you for your time and attention.
Maybe we have time for just like 2 questions. I know you had one earlier.
That was interesting. Thank you. I was going to make a comment that I thought maybe you could counter-comment on. So for educators thinking about data like these, one of the most obvious connections that people can make, which is probably way below the level of thinking of that's going on in your lab, is that more activity in the brain and even more tissue in the brain isn't necessarily better for kids. It doesn't necessarily go along with better outcomes. But yet in the colloquial conversation about brain development in education, we think about more activity, getting neurons to fire, more things happening is always better. So yeah, I thought maybe you could make a statement to that effect, but sort of interpreting it through the lens of pulling together the different syndromes that you're talking about. You don't have to, if I'm putting you on the spot.
I just agree entirely. So, we live in America and bigger is better. It's obvious, right? And it's not necessarily the case at all. And there's lots of other data we have in the lab and that have come from other laboratories suggesting that the brain has to do important work in development, and part of that is eliminating unnecessary connections that early on may be very helpful. So young kids are very good at learning new tasks, so their fluid intelligence is very strong. Adults, not so much. But adults, when you find your ecological niche, so when you decide you're going to be a teacher, like my daughter, she's going to be a monk and poor the rest of her life, but she's in education now in Delaware, and so she's found her ecological niche, and so all the other stuff that was in her cortex, all those other connections, she doesn't need so much, right? So as she becomes a better and better teacher, she needs to activate and maintain those circuits that do support teaching, and you become better and better at that. So crystallized intelligence is much stronger in adults typically than in kids. So it's this trade-off developmentally, and it has its basis very much in the structure of the brain and the connections. And then neuroplasticity overlays on top of that maturational trajectory to sort of fine-tune things. It doesn't change things completely. It fine-tunes what's already there and maturationally driven.
So we actually need to call it Untold. So
This for the class coming in.
this class
Sorry
coming
about
in.
that.
So you can maybe ask some questions after. Thank you very much again.